Okay, now it's 30 each. 50% is attended. Okay. Already the cell theory, cell structure, cell constituents, and evaluation of biology, engineer and biology relations, introduction of biology, everything is over. Today's is our class up to 4:50. The 2 hours class is according to the new timetable. Actually, today's our class is lab, they mentioned. The first 1 hour we discuss some theory, the next we go to the lab. But today is up to 4:50. Don't escape. Okay. Next, we have another 3 topics in our unit 1. One is molecular taxonomy, carbon-based energy. The next is in cellular life. Cellular life. What is the cellular life? And at the same time, prokaryotics and eukaryotics differentiation. This thing definitely you can cope up with that in our entire the cell and cell biology and cellular life of living things. And this lot of times prokaryotics and eukaryotics and their differences you can easily distinguish. Okay. Why is the cellular life? It just begins either in animal kingdom or the plant kingdom, the biggest kingdoms. Even though the protista is also, but it's in a bacteria, eubacteria. In all these, the millions of organisms is identified, and their cellular life is in what way it begins. Someone is the microscopic one, the old theories of cell theories, and the new era, the new cell theories is generates. So the cellular theory accordingly, the division of the cell growth, and the uniformity of the cell, in what way it's
Sir?
leads by genes, the DNA, the chromosomes, all these are in what way its influence of cell growth. We identify in this the cellular the life, cellular biology, what it is. Okay, only 44 students, including me. Yeah, tell me.
So according to our timetable, we have class only till 4th, sir.
4.
Yes.
But it's a new. What about the next hour? Cheppamma?
Free, sir. Very class later.
Is it any subject? Is the next hour?
It's free, sir. We don't have any class.
Free, yeah. Free
But
means.
according to our timetable for life science, we have class only till 4.
And the new timetable I got it the last week. There they mentioned scattered of our 2 hours up to 4:50. They mentioned as a lab, life science lab. So that is the reason why I code it here up to 4:50. I create the session like that. You have a free class means you shall no bother about. You have any class, the SAR is its may, he creates the class, then we think about that. Isn't it?
Okay, sir.
We will do. Here, this is one theory. Yeah, 2 theories, 2 lab hours and 2 theory hours in the selective subject. 2 lab hours and 2 theory hours. So in our the total 4 hours, they mentioned on the Monday for your timetable, the Monday and the Friday is in the 3 hours. And the Tuesday is from 3 to 4:50 is lab hour they mentioned. But that new timetable, I think you may not reach. Now that link is also not yet open. Every faculty members also suffers to that one. But is it really it changed or not? Some of the branches of the students, they don't know. They are not yet joined. Whatever the little bit of changes in the total 4 hours, they don't know. It is all of a sudden they are given. But now the same link is not yet open. Okay, anyhow, you have the free hour. Definitely. We try to. Why? Because of our exams is on first week of August. Some slowdown of our syllabus lag behind. And of course, one is in biological science, is it's life science. For engineers is like that. But we do it's in a favorably. Okay, the cellular base of life. I will share you. Is it visible? Now it's 50 participants, including me. Okay, is it visible?
Yes, sir.
Yeah. Cellular base of life. What is the cellular base? The cell is a unit. The cell is a unit for any creature. Is it in a unicellular body or a multicellular body? The functionalities, systems, everything is develops through this one only. But it's in a what way? The several golden theories, the new era, so new theories is also develops. The cellular is nothing but of the communication. The cellular it's a biological based word. It is a cellular base of life. Cell and its division in what way? What way it is accompanies to the rest of systems which it adapts in this way. In our introduction, we have a lot of the great scientists and everything we know about already. But anyhow, these are the things in front of us. What we know or unknown, someone is an average human being is composed around 100 trillion individual cells. It's great. And human blood is also passed in a day. How many kilometers? Just you assume. Just like it's in the pumping blood is pass on, it travels even a small vein too, the biggest nerves too. In what way? Just you image to that one. Yeah. Next, each cell has its about 10,000 times of the many molecules, just like it's a Milky Way of the stars. This is the another thing. And 300 million cells. You see here it is. This is a unknown factor. Is die in a human body is in every minute. In what way? What way? We know about our circulatory system contains of the red blood cells and white blood cells. It is antigens and antibodies is fight outs. Of course, the white blood cells it dies, and at the same time the red blood cells is also deteriorates, and the new one is originate in the bone marrow. This is the thing of circulatory system in the human body and all mammals. This is the thing. But this decade one is surrounded by the lysosomes, just like it's a burial ground. The lysosomes can lysis to this, all these particular cells. Yeah. The discovery of the cell, the great scientist of Robert Hooke. In introduction class, we are already identified these great personalities. When he discovered the cell while he's looking of the thin slice of the cork. Yeah, he discovered this cell in the tiny boxes. The tiny boxes, just you think about the last, in the microscopic laboratory, the microscope lab, I think it's over. There is in a plant cell, there we are identified the rectangular shape of the boxes in the chlorophyll, or else somewhere is in a, just like it's in a honey chamber. The honeybees, nest of the honeybees in what way? It's like that it seems to be. He mentioned, he thought that cell is only existed in plants and fungi. He identified up to that. It's the old technological, the microscope. And the next is Antony van Leeuwenhoek in 1673. He used the handmade microscope, the microscopic way of organisms he identified to observe the pond's scum, the pond water, freshwater pond. The pond scum he observed, he discovered the single-celled organisms. So many single-celled organisms he identified. It's called as animalcules. It's called as animalcules. Already this word you are identified. The animalcules, the protozoans is animalcules. So that is the reason why it's one of the organism of paramecium. It's called as a slipper animalcule. Why? Because its shape is in this slipper-like. Yeah, and he also observed the blood cells from the fishes and birds and frogs. Of frogs is amphibians, the birds is its aves, the fishes is fishes, and the dogs is mammals, and human. He observed to that one. Therefore, it is known as the cell is found in the animals as well as plants. In both the cases, it's a lot of developments of theory. So the originated of the body is unicellular or the multicellular is used by cell theory. The cellular divisions, cell body, cell through the body generated the great scientists in 1838 and 39, the German botanist and German physiologist. It's both are they identify. One is in a plant parts, and at the same time, this Theodor Schwarzmann, who was the close friend of Schweldring. These both are the friends, good friends. He started to that all animal tissues and composed to the cell. These both are identified. The next, the development of the cell theories it begins in 1858 onwards. The both plant and animal cells, whenever the similarities is identifies, the next the cell theory just begins. Then Rudolf Virchow. He is German physicist. He identified, he extensively the study of the pathology. Cellular pathology. The pathology is nothing but the disease-caused organisms. He concluded that the cell must arise from the pre-existing cells. Whatever these existing cells do to this, is in a diseased one or else some abnormal growth or whatever. He identified through this. The next, three basic components of the cell theory. The cellular based theory is in from 1838 to 39 up to 1858. He identified three bases of the cell. Definitely, these 3 points were already discussed. The organisms are composed in one or more cells. Okay. The cell is its basis of the unit of the life, the living things, and all cells is produced by the division of pre-existing cells. Yeah, this is the fundamentals of the basic points of the cell theory. What is in a modern cell theory? This is totally the genetical way of thinking. The modern cell theory contains of the 4 statements. In this, the addition to the original cell theory, the cell contains the heredity information in DNA, deoxyribonucleic acid. DNA, the hereditary characteristics, is its pass on to the next generations, which is passed on from the cell to cell during the cell division. Cell division is it's in mitosis or meiosis, but the information is passed on through the DNA containing of the chromosomes. This double helix structure of DNA containing of the chromosomes, the pairs of the chromosomes through pass. All cells are basically some of the chemical composition of the metabolic activities. We will read of the next. What is a chemical component? The cell, the DNA containing chromosomes containing proteins. Those are adenine, guanine, thymine, cytosine. These are the chemicals is majorly the influences of cell components, cell compositions, its influence. All basic chemicals and the physiological the functions are carried out inside of the cell. There is in a movement or digestion, means it's destroyness also. Yeah. And the cell activity depends on the activities of subcellular structures within a cell. Yeah. Already we know about the nucleus, the plasma membrane, and organelles. A lot of organelles is inside of the cell. Each and every organelle is an individual functionality. Yeah. Okay. Anyhow, these are all the modern cell theories of the four statements. It's a totally the hereditary way of the functionality is based on it transmits to DNA. The DNA is contains of the nucleus you know about. Okay, this DNA is in both single cell means unicellular and multicellular bodies. In both the cases, both prokaryotics and eukaryotics, this DNA is possible. So now these 3-4 statements is satisfied with the modern cell theory. It's satisfied. Okay, already in our laboratory experiments of microscope, we identified several types of microscopes, the magnifications, the light microscope 400 to 1000 x megapixels, it's magnifying 1000 times. A confocal microscope, fluorescence microscope, on the other hand, it's 500 x, and electronic microscopes 1000 to 10,000 we can identify. Yeah, these three, SEM and TEM. They're even small hair-like, human hair-like structure is also small tissues, very micron, the nanometer size is also we can accelerate, we can identify through this. Why? Because the electron microscope, it can up to 10,000 times it can magnify. Yeah, you see, this is the microscope parts and all these objective lens, optic lens, and all this. I think the microscope laboratory is our. Hello. Hello, no one, it's not an answer. 52 members. Microscope lab. Already I show the microscope lab, draw the microscope and all these are the last week. Hello. Vidya, Sri Vidya, 56, Sri Vidya, 57, Nikit, Sai Shivani, oh, one can't respond. Manish Reddy, Manish, Diksha, GB Diksha, 22, 26, 30, Siddharth Ravi Kumar. Oh, all are just you opened it and went out. Number two, Abhishek, number 24, 24.
Yes, sir.
Yeah, you are only present. But the rest of people, so many people I called. Abhishek, what about our Abhishek? He is also class leader. Emma, number 2. Abhishek is one of the leader, isn't it?
Yes, sir.
అభిషేక్ ఏంటి ఎంతసేపు పిలుస్తున్న పలకటంలో లేదా నువ్వు లేవా అసలు అక్కడ మైక్రోస్కోప్ ల్యాబ్ చెప్పామా లేదా లాస్ట్ వీక్ అభిషేక్ నెంబర్ 2 24.
Yes, sir.
What about the microscope? The lab is
Hello?
over or not? Last
Yes, sir.
week? Is it over? What about the rest of people? Why these all are? No one can't respond. Bhargavi, 39, Bhargavi. Vamsi. Surya Satvik. Nikhil. 42, Nikhil. 43. Who is 43? 48. All these are the participants list, but no one can't respond. Sharath, number 3, Sharath.
Yes, sir.
Sharath, ఏంటి నువ్వు నిద్రపోయినట్టు మాట్లాడుతున్నావు
No, sir.
మరి ఎంతసేపు పిలిస్తే ఎవరో ఒకరు పలకచ్చు కదా
చెప్పండి సార్.
ఇప్పుడు మనకి మైక్రోస్కోప్ ల్యాబ్ అయిపోయిందా కదా చూపించానా మీ మైక్రోస్కోప్ మీరు డ్రా చేయాలి అని అనేసి అని చెప్పాను కదా అప్పుడు వినిపిస్తుందా
ఎప్పుడు సార్?
లాస్ట్ వీక్ అసలు మైక్రోస్కోప్ చూపించాను హౌ టు డ్రా ద మైక్రోస్కోప్ టైప్స్ ఆఫ్ మైక్రోస్కోప్స్ గుర్తులేదా లక్ష్మి అఖిల
అవును సార్ చెప్పారు.
ఏంటి అవును సార్
I mean like last class చెప్పారు అంటున్నాను సార్.
చెప్పారు. ఓకే.
ఎస్ సార్.
చెప్పాను కదా, అది అందుకోసం అంటే మళ్ళీ ఇందులో మైక్రోస్కోప్స్ వస్తుంది, అవి మీరు వినే ఉంటారు అది.
ఎస్ సార్, ఎస్ సార్.
ఓకే. ఏంటి ఇంతమంది కూడా అందరూ 3 o'clock ఏమైనా పెద్ద మీకు ఇంటి దగ్గర ఉండి కూడా ఎవరు క్లాస్లో అందరూ
ఉన్నారు సార్.
ఉన్నారా?
ఉన్నారు సార్, అందరు ఉన్నారు, అందరు వింటున్నారు.
ఓకే. మీరు ఫెయిల్ అయితే ఇట్స్ షేమ్ టు మీ. మీకు కాదు, నాకు షేమ్, నేను షేమ్ఫుల్గా ఫీల్ అవుతాను. మీరు
అసలు ఎవరు ఫెయిల్ కాదు సార్, పక్కా సార్,
ఎవరు ఫెయిల్ అవ్వకూడదు. ఓకే.
ఓకే.
The microscope already I will send you that videos also. Anyhow, this microscope, parts of microscope, how it is we handles to this one, in what way it magnifies. These are all the several types of microscopes we are identified. Yeah, these are all compound microscopes. It enable to us to see the overall shapes and structure of the cells. These are the cells. You see here it is the structure and the morphology and the function. The function of the cell we can identify through this. It's confocal and fluorescence microscope through, because it's the 3D. It's a 3D, the 3D detection of the biological object to the labels. So that is the reason why we can confocal microscope through both structure, morphology, and functions. We can identify HU and controlled and highly limited depth of the focus is possible through this one. The next is very advanced one in the research laboratories, where we are using of SEM and the TEM, scanning electron microscope. And this both are electromagnifying through the magnifications. Here it's also 3D structures we can possible, but even the hairline structure also we can transmission and scanning. Through this we can identify it's very the sophisticated ones. These are all are lakhs of rupees of cost. Now, derivatives of the cells. Just you identify this the cell structures of the skin cell, the bacterial cell, and at the same time the chlorophyll. This is a plant cell, the chloroplast, and the egg cells, and the nerve cells. This is dendrons and everything. But this one, red blood cells, and this is the plant tissues, and this is what it is. This one is its sperm cell. The sperm cell. To this flagella, it moves and pass on to reach the egg contents of the ovum. It hits and then it is fertilized. Okay. That is another functioning of the role, of course. These are through scanning electron microscope through prokaryotics and eukaryotic cells. The both. Here it is eukaryotic cell nucleus, but here it is direct DNA, nucleoid, it takes place. Okay, nucleoid. It's the no cell wall. This is the major difference. The two fundamental differences of the two types of cells, prokaryotics and eukaryotics. Here you see in presence and absence of the nucleus. One is already these are all well, prokaryotic and eukaryotics. The direct DNA, here it is. Okay, and both the cases of cytoplasm and the plasma membranes, ribosomes, all these are, but size wise, 0.12 in the 100. It is 10 to 100. Prokaryotics and eukaryotics. The major domain of the prokaryotic cell is we can exemplify through as in a bacteria, Escherichia coli. Escherichia cholera, one of the disease causing the bacteria epidemics. A lot of the epidemics is in, it can spread out to this Escherichia coli. So water bodies, the water bodies is contaminated. Any fecal stands, it may combine with drinking water then immediately. This is, and the Staphylococcus. This one is also one of these round shaped bacteria. Yeah, a lot of bacteria. It's a good example. It's a good domain of prokaryotics. A singular cell is a no nucleus, no components. It is the peptidoglycine cell walls. This is totally peptidoglycine cell walls. It can breaks easily. It can easily penetrate. It can easily attacks to other bodies. The hosts, it can easily enter into the host. The binary, the fission for energy and use organic chemicals and inorganic chemicals for photosynthesis. This is a lot of this bacteria is a chemobacteria is also. We can identify this is a chemosynthesis. Photosynthesis we know well about that particular word. Photosynthesis is in presence of sunlight, the photons of energy in presence of carbon dioxide and water. But the chemosynthesis, this is the thing we can identify the next the topic of our carbon-based energy. The carbon-based energy, in what way? It's a chemobacteria. The chemosynthesis takes place. These are the things we can identify. This is the energy source. It's in organics or inorganic, whatever. In a chemosynthesis majorly, and the photosynthesis is a commonly. Some of the Anabina is a bacteria, the cyanobacteria. This Anabina bacteria is very helpful bacteria. Here it is, but it is a Escherichia coli. It is an effective bacteria, disease causing one. But Anabina, Nostoc, all these are helpful bacteria in nitrogen cycle in our the laboratory experiments. One is the nitrogen cycle, nitrogen fixation, nitrification, and all these are we are identifying. There it is. Anabina bacteria, it's a cyanobacterium in what way? But it's not all these are the maximum. The maximum size is in the 10 micrometers under microscopic conditions only. Yeah, it's a prokaryotics. The domain is in archaea already. We know archaea is in both the cases. Already we know both autotrophs and heterotrophs also. It leaks peptidoglycine. It leaks. It lack of It's lack of peptidoglycine. The domain in this particular archaea, the domain. It's living in extreme environments includes methanogens and extreme halophiles and extreme thermophiles. Highest temperatures, lowest and deep conditions, and in a deep earth also. For example, this bacteria's load we can identify in a what it is, mines, coal mines, especially the coal mines, we can identify. The role of disease is not well understood. This group has its only recently been discovered, but this particular domain of archaea containing of prokaryotic, but it's a unicellular one. But here it is the difference It is in a lack of peptidoglycine. This peptidoglycine layer is missed. Next is eukaryotics. Is the developed one. One is the plant cell and another one is animal cell. In both the cases, it's the biggest vacuole is contains of plant cell, but here not that much of vacuoles is not that present in the smallest vacuoles somewhere is in a nucleus, ribosomes, mitochondrions, everything is in a possible. Yeah, the major difference is the chlorophylls is contains in the plant cells. Yeah, the structural differences, plant cells is in the chloroplasts. Yeah, chlorophyll containing chloroplasts as a large centriole, the vacuoles as a cell wall, and the plant cells do not have any centrioles. The plant cells have in a plasmodesmata. The animal cells have gap junctions. This is the smallest differences varies in between. This is the plant cell. This is of our animal cell. The physical differences, the plant cells have in a photosynthesis in addition of the respiration. Respiration is also takes place in the night times, during the night time. Yeah, during the mitosis, a cell plate is formed in plant cells. The starch is a molecular for energy storage. Yeah, after completion of the photosynthesis, the starch, energetic substance is generated. The animal cells is in a glycogen substance. Yeah, the large centriole vacuoles, the store and the more water and carbohydrates than in the animal cell vacuoles. The biggest vacuole is takes place in plant cells. Here it is not that much of, just only the gaps is takes place. This major differences between plant and animal cell, but this both are comes under eukaryotics. Eukaryotics. You see, you can go through easily identify this particular the diagrams. Is fully clear. The difference in between the plants and animal cells and the various specialization functions occur in a specific places. These places are called originellas in a small organs. Yeah, this organs is differs. The first foremost one is in the chloroplast. Yeah. Yeah. Next is the plasma membrane. The structure of plasma membrane is the proteins, the carbohydrates. These all are one to another is attached in outside in the particular. This is total the cell membrane, the lipid bilayers, two layers, lipid bilayers. These protein channels, it can releases, it can stop inside of the cell in a cytoplasm. Just like the cell wall, it acts as a gate. Already we are identifying the fluid is a plasm from outside to inside. Next is the cell membrane. It separates the living cell from the non-living surroundings. The thin barrier, 8 nanometers of thickness. It controls the traffic in and out of the cell. Already in yesterday's class, Monday class, we are identified. It is selectively permeables only. It's a good one or harmful one. Accordingly, it can allow some substances to across the more easily than other. The hydrophobic or hydrophilic. It's in attracts towards the water or it's in a opposed to the water. Hydrophobic and hydrophilic. It's made up of the phospholipids and the proteins and other macromolecules. Yeah, this is the phenomena takes place This is our hydrophobic, but these are all are hydrophilic. There is, this protein portion is a bilayered one, hydrophobic and hydrophilic structures. Next is the membrane. It's total, it's in the proteins wise. Yeah, extracellular fluids and cytoplasm. These both separates through this particular the membrane. The membranes can transmit in a different way of cell components. Glycolipids. The glycolipids it packs out either in carbohydrates or proteins. Yeah. And phospholipid bilayer. What it is? These are all water soluble one. These are water insoluble one. These lipids is organic compounds, fats and oils, and the non-polars. It's not in water-like. It's insoluble in water, but not attached to the water. This is the pot, and the phosphate head is in a pot, polar, water soluble is attracts towards the water. The phosphate group and the glycerol backbones. Inside and outside containing of the cell walls. It's in hydrophilic and hydrophobic through these particular bilayers can separate. And permeability of the phospholipids in bilayers. What? The carbon dioxide, benzene, ethanol, water, and the glucose, amino acids, and other ions of calcium, nitrogen, sodium, chlorine, hydrogen. In all these cases, depends upon is in hydrophilic or hydrophobic. This is a fundamental, in our chemistry. The internal composition of the cell is maintained because of the plasma membrane is selectively permeable to the small molecules. Either it's in a polar or the non-polar ones is based on to that it's eliminate or withdrawal or active to allow. Only the small, relatively the hydrophobic molecules are able to diffuse across this particular phospholipid layer, this particular phospholipid bilayer. Bi means here it is 2. Phospholipid bilayer at in significant rates of using and the passive diffusion. You know about the diffusion, osmosis, transmutation. All these are we go through. The transport across to this particular the membrane in what way? From outside to the inside. Simple diffusion. This is a simple diffusion. This is a channel mediated diffusion. It's a carrier mediated diffusion. Yeah. This is the transported molecules. The channel creates, and at the same time, it is a passive transport. It's an active transport. It's automatically, but here it is active transport. Some energy, some external energy it requires through active transport from inside to outside or outside to inside. The concentration of the gradient. The gradients is in low concentrated one or high concentrated one. But anyhow, the transportation across the membrane is in a passive transport or active transport. Especially this entire thing we are discussing the cell membrane through this simple diffusion the channel through, and carried moderate. The transport across the membranes is in a passive diffusion. We know about the diffusion. The movement of the molecules or ions in a region is where they are is in high concentration here. The high concentration to the region of the lower concentration. It diffuses. It diffuses towards. This is of the transport membrane through. This is the passive exercise of transportation. Here it is. The area of the high concentration, the area of the low concentration, it can pass. It's a simple diffusion. It's a facilitated diffusion. It can allow. It's allowed. Then only it diffuses through the particular channel. Diffuses through carriers, the catalysts. It can pass on. Facilitated. But these three is in the passive transport. There's no need to of energy. The energy is required to the active diffusion only. The simple diffusion, for example, the gases like carbon dioxide and oxygen, and the liquids like aromatic liquids like of our hydrocarbons, it's a benzene, it passes on. The gases and the water molecules, its rate is slow due to the polarity. The water molecules, the passing is through polarity. One is the negative pole and another one is the positive poles. Both is takes place. Yeah, the lipids, steroid hormones and the lipid soluble molecules, hydrocarbons, alcohols, and some vitamins. These can easily it dissolves. It passes. And a small non-charged molecules, ammonia like, you have no charged one. Ammonium ion, NH4 plus, that is the charged one, but here it is ammonia, the molecule of ammonia, NH3. Ammonium ion is different, it's ammonia. These are all it's in a simple diffusion, it can simply. And the facilitate to diffusion. The way it generates, it can slowly, but need not any energy, external energy, but extracellular fluid to intracellular fluid means cytoplasm, it can reach us through channel portion. Yeah. This is ions like sodium, potassium, calcium, sugars like glucose, amino acids, and the small water soluble molecules also, and the water in a fast rate. Yeah, it can carrier proteins and immediately it closes. And next, how to do that? These are all of the cellular, the life, the cellular life of living things. This is a major functionality. Then only the cell is growth and functioning is carrying way. It carries in a fruitful way and a cohesive structure it can generate. That is the reason why the cellular, the life, cellular body agents. How do the molecules is move through the plasma membrane? By facilitated diffusion. This is the thing. The channel or it's in a carrier, the proteins are specific and the channel proteins is yellow ions and a small solute or in a water to pass, water soluble sponges. Okay, the carrier proteins is moved to the glucose and amino acids and facilitated to the diffuse to the rate limited by the number of the protein channels or carriers is passed on to the membrane. Here it is have no any external pressure. It's totally active transportation. Active transportation into the membrane. The next is osmosis, osmotic pressure in your water lesson in your engineering first year. All of you know, I think. Someone is left out, 2 members, now it's 50. Students, 49 only, including me, it is 50. Okay, osmosis. Osmosis is diffuses of the water across the differential, the permeable membranes, yeah, through osmotic pressure only. osmotic pressure. In the pressure that is develops in a system due to osmosis. So osmotic pressure only can pass on. Yeah. The water molecules, it's a permeable membrane, it's a differential permeable, and the membrane it can allow, but at the same time, through osmotic pressure only, it reaches towards concentration of the water. Here you see this one is in a hypotonic and hypertonic. Yeah, hypertonic, hypotonic, and isotonic. In all, these are the solute and solvents differentiation. In general solution, always the solvent is very high and solute is very little bit. But in case of, that is a general phenomenon. In a sugar water, the sugar is in a solute, is in a small quantity, and a solvent is water. Now these both are mixed up, there is sugar solution. Here, the hypertonic is more solute, the less water, less solvent. Hypotonic is less solute, the more water, solvent is its high and solute is its low. And isotonic, is both are equal, both solvent and solute is equal. According to the concentration of the water, the direction of the osmosis is determined by comparing of the total solute concentrations. Yeah, wherever it is beyond the level of the osmotic pressure, then we create the external pressure. Then only it can. One is in hypotonic, but this one is in hypertonic. It's more water. Is hypertonic, and it's a more solute. But here it is, its net movement of the water is pass on from one end to another end through this permissible the membrane. The osmotic pressure is beyond the level of osmotic pressure wherever it's applicable. Then the reverse osmosis, that is the reverse osmosis. But this is general osmotic pressure, it's only it can pass on. The next is active transport. Active transportation, ATPs, energy, and all these are, we will discuss later. Adenosine triphosphate, you remember, whatever the food material is prepared to photosynthesis of these carbohydrates and everything, this energy is in a form of ATP, this active transport. These things we discuss this later. But anyhow, this is of our cellular, the basis of life. Cellular basis of life is depends on the cell and cell wall and cell constituents and their growth and functionality. Okay. So don't quit from this class. We have up to the 4:50. What he shot. Sir, I am present. There is issue in my mic. Who is he? Abhishek, number 2. Okay. No problem. Don't scatter. Please, you have attendance is also calculated up to 4:50. Our class is 2 hours. The next hour is also for us. The new timetable. Okay. The first lab is virtual lab of microscope, different types of microscopes, the drawing of microscope. It's over, I think. How many people is draw the microscope? On that day, I have no time, I think. Not yet seen anyone. If anybody draw the microscope in a rough paper, even the minimum in a rough paper, if anybody please show me microscope. Anybody draw that particular microscope? Compound microscope. Out of 49, no one. Anyone, please show me. Who is he? 25, 25, 25, Bharatamata, photo పెట్టారు. 25, Spider-Man, 28, 28, who is he? Change the Spider-Man photo. Who is this? 006, 1006 on the wrestling. Is a wrestler or some WWF? One 1047, 1047. Okay, anyone to draw the microscope? Please show me. Cycle. Can I start the nitrogen cycle? Can you draw the microscope? If you may draw the microscope, I start a new virtual lab of nitrogen cycle. Other than that, definitely I will show you the microscope, how to draw the microscope. You see. Please take one rough paper or book. White pages, 100 pages, white pages book is needful
Okay, sir.
for our virtual labs. 100 pages,
Right.
white, long notebook. The next, to forward the next lab, virtual lab of nitrogen cycle. We start today. Okay, all of you quick draw this microscope. Abhishek, నువ్వు చూపించాలయా?
Okay, sir. Okay, sir.
You are a leader.
Sir.
Class leader. Class leaders first show your performance. And the 24 Priyanka, 24 Priyanka, you also be ready with
Sir,
this
by
is the base.
next week lab on. Sir.
Base structure of microscope, handle of the microscope, It's easily engineering drawings like can. Whatever the shape you may occur, it's no problem. You just once you show me. All of you show this one. Please draw the microscope. Even a small paper. Next, you record it in your observation book. These both are fine adjustment and rough adjustments.